Hi. Well, welcome to Unit 5. You'll notice when you notice later the background on my screen, you're going to see stained cells of onion root tip. An onion root tip is used quite often in schools to demonstrate stages of mitosis. In this unit, we're going to talk about reproduction, both asexual and sexual. Today we're going to talk about asexual reproduction, which is primarily a discussion of mitosis. We need to understand that before we can talk about sexual reproduction. You're going to find out that you probably didn't know very much about sex at all. And you're going to learn, I hope, how very, very interesting sex can be, but not in the way that you're thinking. Get those thoughts out of your mind. We're going to talk about meiosis. But today, right now, in this first video, I want to talk about mitosis. Mitosis is the process primarily of asexual reproduction. And it happens millions of times every day inside your body, in most of the tissues of your body. Cells die, and they need to be replaced with new cells. And so let's take a look at what actually happens in mitosis. The cell cycle is represented quite often in a circular diagram. The problem is that's not always the best representation, because what happens is when the cell cycle is over, we have two new cells, and the cell cycle may or may not start over. We could look at it as a straight line continuum, but this animation, which is a pretty good one, is available in your PowerPoint, so let's take a look at it.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing two copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has four stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker.
So I want to pause this for just a minute and let's back it up. One of the things that people forget to tell you about, let me just go back here, in interphase, which takes most of the time of life of the cell, this is when DNA replicates. Well, that's what they said, but there's so much more that goes on. The cell grows here, and as we learned back in Unit 2 or 3, there are a whole bunch of cellular organelles: mitochondria, Golgi bodies, different membranes, ribosomes, rough ER, endoplasmic reticulum and smooth endoplasmic reticulum, and cytoskeleton and others. Well, all that has to replicate too. And during the growth phase or interphase, all of those cellular organelles replicate. So it's not a resting stage. It's a very, very active stage. And that's the point. And then, of course, he talks about the 4 phases of meiosis, which is prophase, metaphase, telophase, anaphase, and telophase. And then, of course, once this is done, and this has to do, these phases, this part that's mitosis, has to do with replicating all of our DNA, our genes, so that the next cells that result from this have identical genetic material in them. So they are basically clones in mitosis. Mitosis produces clones. It produces new cells. And this can continue for about 50 times, and then it's over. So when most of the cells of your body have replicated themselves about 50 times, you die. And that's the end of life.
During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase. And in these phases, duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
So we have 2 identical daughter cells, but it's more than that, isn't it? How is it that if we start as a single cell organism and we start dividing our cells by this process called mitosis, that we end up with liver cells, skin cells, muscle cells, and all these different kind of cells? Nobody knows. That is a mystery. But that's an important concept and an important mystery, because it is this mystery that people are using today to do new types of science in the medical field. They are looking at stem cells, which are the cells that do this initially. And many of our tissues have their own stem cells. We have stem cells in our bone marrow that produce multiple kinds of white and red blood cells. We have stem cells in our brain that produce new brain cells, and in other parts of our body. And so, what people are doing is they're doing research using stem cells to see if they can't use them to produce new cells in our body to replace disease cells. For example, it is hoped that someday stem cells can be used to produce new cells of the pancreas that are responsible for producing insulin. Because many people today are wearing out their beta cells by eating too much sugar, and they're developing type 2 diabetes. And other people can almost be born with type 1 diabetes, which means there are no beta cells left at all to produce insulin. So perhaps by injecting stem cells that do mitosis into organs that are diseased or defective, maybe is a better word, perhaps we can then develop new beta cells to replace the ones that aren't functioning. We don't know how that happens, but we know that it does happen, and that's the important thing. Now let's look at homologous chromosomes. We have 23 chromosomes in each of our cells, in every one of our cells. And that means that our body has a lot of DNA in it. The purpose of the strawberry lab that is attached to the content tab here that you will find is to show you how much DNA there really is. There's a lot. In this case, the homologous chromosomes are the ones that are one from mom and one from dad. They produce a pair. When these replicate, we have sister chromatids. Here's the dad's chromosome, the one you got from your father. No, the one you got from your mother. Purple is from your mother. And it replicates. And here is the father chromosome, and it has replicated. And then we see that we will get, every one of our cells will get one of our mom's and one of our dad's chromosomes, because these will pull apart. They will line up and pull apart. During mitosis, these will line up on the central plate during metaphase, and they'll be pulled apart, so one goes to one new cell and one goes to the other new cell. Okay, so I might have to go back here. That's the one I want to look at. We have 23 pairs of human chromosomes. Here they are. 23 pairs. One of the pairs, number 23, which is right here, I'm circling around it, has an X and a Y. If you have two X's, you're a girl. If you have an X and a Y, you're a boy. If you have three X's and a Y, wait a minute, how does that happen? Well, it does happen sometimes, and that's called a chimera. A chimera is the fusion of two zygotes. It happens in early embryonic development, very seldom, but it does happen. What it results in is two people in one body. And so a chimera can be 4 X's, or it can be 3 X's and a Y. It can be also 2 X's and 2 Y's. So it can be the fusion of two girls. It can be the fusion of two boys, if there's 2 X's and 2 Y's. Or it can be a fusion of an X and a Y with an X and an X. In other words, a girl and a boy could fuse. This chimera is a true hermaphrodite. It is not an anatomical hermaphrodite, having both sexes. It is a genetic hermaphrodite and a true hermaphrodite. Interesting, don't you think? There are recorded instances of people who are true hermaphrodites, and I wonder if Annie Oakley was, because Annie Oakley was a very unique individual, genetically as well